In this short tutorial we're going to focus on just a few of the different ways that transgenes or genes can be inserted into patients for gene therapy. This is an attempt to rectify some kind of genetic abnormality, some genetic problem where a particular gene isn't functioning properly and therefore a particular protein is not being made that leads to some kind of genetically related condition. So if we can fix up the faulty gene by replacing it or altering it in some way, then we can hopefully go some way towards fixing the condition that the patient has. There are a number of different ways that these transgenes, that's a gene that's been made to work properly for gene therapy, can be delivered into a patient. They can either be put directly into something like a virus, like an adenovirus, which we'll have a look at in just a moment, and be injected directly into the patient so that the virus then delivers the gene directly to target cells. Or it could be that you've got a cell-based delivery system where the gene is likewise packaged into a virus, and that virus then injects the gene into cell tissue outside of the body, and then something like stem cells, and then these stem cells are introduced back into the body and will then differentiate and work properly. So there's a couple of different ways just there, and we'll have a look at a couple of others in just a moment. Okay, we'll just have a quick look at how an adenovirus might be used in this process. So if we look at this diagram here, you should be able to remember from Unit 3 how viruses basically go about reproducing. And they do this essentially by injecting their own nuclear material, their DNA or RNA, into the nucleus of the host cell, and then the host cell will go about transcribing and translating that viral DNA. And in normal viral reproduction, this would lead to new virus particles. But it's possible to engineer viruses so that they don't reproduce themselves, but they can still inject their DNA into a host cell. And in this way, it's possible to deliver a transgene, that is a gene for gene therapy, into a host's cells, and therefore with some hope that the correct gene will then produce a correct protein and overcome a condition. So just a quick reminder of how this works. Now this is looking at the use of an adenovirus vector. So we can see up the top here that the DNA, or the gene of interest, is modified so that it can work correctly. This will be done in the laboratory. The gene of interest, which could be a human gene, is inserted into viral DNA using recombinant DNA technology, which you've studied. So the new gene's inserted into viral DNA, and then it's injected into a viral vector. Now remember, the word vector means something that will be able to deliver something into something else. It acts as a transport mechanism. So the modified DNA is injected into an adenovirus, which then is delivered into the patient, and when it comes across the host cell, in this case the adenovirus will be taken in by endocytosis and encapsulated in a vesicle. This vesicle will move into the cytosol of the cell and then break down, releasing the viral vector into the cytoplasm. The virus then injects its modified DNA, in this case, into the host cell's nucleus that you can see here. These are the nuclear pores that are seen in this diagram. And then there's modified DNA, which contains the gene that we have originally inserted into the viral DNA, will now be able to be transcribed and translated into the protein that is needed by the patient. So this is one way that an adenovirus vector can be used to inject a gene into a patient to get them to produce the correct protein. Let's now have a look at how a retrovirus can be used to insert a therapeutic gene into a host cell. A retrovirus is a virus that contains RNA as its nuclear material. So the virus particle contains RNA, not DNA. And so, as you could probably work out by now, and certainly from our work on viruses, you would have to realise that a retrovirus will have to turn its RNA back into DNA inside the host cell so that that DNA can be inserted into the host's DNA. So a retrovirus has to convert its RNA back to DNA. So a retrovirus is an RNA virus, not a DNA virus. So when it enters a host cell, it also injects a very special enzyme, which you've also heard about in this course, called reverse transcriptase. Which of course is used in other lines of genetic research. So the virus injects RNA plus reverse transcriptase into the host cell. The reverse transcriptase turns RNA back into double-stranded DNA. Now you can see this up here in this diagram. So there's your single-stranded RNA from the virus. Reverse transcriptase enzyme converts it back into double-stranded DNA. The double-stranded DNA then gets inserted using a couple of other enzymes into the chromosomal DNA of the host cell. So if it's possible to insert a therapeutic gene, a gene of interest, into the viral RNA, so that it can be converted along with the virus's RNA into DNA that can be put into the host cell's chromosome. So the gene of interest, and you can read this in a little bit more detail here, the gene of interest is actually inserted into the retroviral genome, into its RNA, and then it gets incorporated then into the host cell's DNA. One of the advantages of this is retroviruses are very stable. They also manage to have a very high rate of production of protein from their genomes inside host cells, so they can be very effective vectors for gene therapy to deal with particular genetic conditions. So you may want to pause this section of the video for a few moments and have a careful read of just the general background on how retroviruses work in general. Just to finish off this tutorial on some of the methods for getting therapeutic genes into hosts in gene therapy, there's a few listed here, and some of these you will have studied earlier in the year when we looked at molecular genetics. So just a quick reminder of what we're looking at. We've just talked about two different kinds of recombinant viruses. That's viruses that have had a therapeutic gene recombined with their own DNA or RNA. So we looked at adenoviruses and retroviruses as vectors for inserting the therapeutic gene into a host. And that can be by normal infection processes, or it could be that the virus is combined with stem cells, and the stem cells are injected into the patient and then incorporated into the body's tissues. So the virus can directly infect the patient, or it can go indirectly via stem cells into the body. Another method we talked about earlier in the year was structures called liposomes, which are little vesicles that are artificially constructed using phospholipid bilayer, encapsulating a therapeutic DNA or recombinant DNA molecule inside it. And these liposomes can be injected into the body and can very easily integrate into the cell membrane, since they have an almost identical structure to the cell membrane, and therefore can fuse with the cell membrane and empty the DNA of interest into the cell, which will then be incorporated into the cell's nucleus and expressed. We've talked earlier in the year about plasmid DNA, so recombinant DNA technology with bacterial plasmids can also be used. And then there are some other methods like the ballistic gene gun, which can insert DNA directly into cells in a patient. So these are all in vivo methods that are methods of introducing a therapeutic gene into a living host. That's what in vivo means. In vitro would mean that you're actually dealing with cells outside the body. In vivo means you're dealing with something inside the body. So these are some of the other ways that gene therapy can occur.